here at the Wilson Museum. We are excited to be finding new ways to connect our collections, our exhibits, and all the wonderful eclectic stories that our founder and people that came after him brought together here at the museum. And how can we connect these to the important conversations that are happening today, to current issues, to understanding across cultures and landscapes and so on. So we've got 4 themes that are being addressed in the course of this Connecting to Collections series, which starting this month and going through the end of the year. And today's falls under our shells section. This is the first of our shells. We had our iceberg presentation that was under the Arctic theme, and we also will be having programming around the stars and the cosmos and around pottery as one of the ways that we can understand culture through the material that people create. And we'll introduce our presenters. They'll talk a little bit about the work, the research that they've done. We'll have some panel questions, and then I'll hopefully allow some good time for questions from the audience. And once again, acknowledging special thanks to Bangor Savings Bank for their support of the Connecting to Collections program series. Haley, is there more? Do you want to say more about the museum in general, or let's see here?
I think that covers it. Though I will add that for those who are close to Castine, who are looking for fun day trips, coming to the museum in the summertime is a really great place to go, especially in July and August. On Wednesday and Sunday afternoons, we have a variety of very active and engaging demonstrations that we offer. So you can experience the sound of iron being shaped in the blacksmith shop, see a boat being built in the wood shop, a variety of different options. So it's a really fun place to visit in the summer. So
definitely,
that's what I'll add.
All right. So welcome to, and I have to take all the credit and blame for the witty title that I come up with for our panel tonight. And it just happened to work out that we were looking for folks that recent research around shells and how shells could help us understand our changing oceans, our changing climate. One of the things that our founder, John Howard Wilson, collected was shells, and we have a display case of shells from around the world. We don't know a whole lot about most of our shell collection, so if any of you are really good at shells, we'd love to have you come, help us learn more. But we knew several folks that were doing work around shells and shellfish that we thought could speak to that, and it just happened that the 3 final panelists all work with, as I've said, the Maya arenaria, or the soft shell clam, which is so ubiquitous along the coast of Maine. So our three panelists today are Kate Pontbriand, who's a somewhat recent graduate of with her master's thesis from the Quaternary and Climate Studies program at the University of Maine, and she's also worked on GIS projects. She currently works in cultural resource management archaeology, and Kate will be talking about her work with soft shell clam shells from a Wabanaki village site in Gouldsboro and contemporary clams from Frenchman Bay, and what that can tell us about oceans and changing use of clams. Sarah Grindle Levesque is a 2014 graduate of the Corning School for Ocean Studies at Maine Maritime Academy, and her research while she was at Maine Maritime Academy looked at the impact of rising sea levels on Wabanaki shell heaps, which are constructed primarily of soft shell clams, and what that's telling us about our changing environment. And then jumping ahead a couple thousand years, Sophie, who's a current student at College of the Atlantic, will be talking about her research into the impact of climate change on soft shell clam fishery and that important part of Maine's economy, and what can be done to mitigate those impacts. So we're going to start out. We'll start out. Each presenter will give a little bit of background on their work, and then we'll go from there. So, Kate, do you want to start us off?
Absolutely. Let me get my slideshow up and ready for everyone. So, share my screen.
And so we did that, and
all right. So thank you very much for the introduction, Julia. I'm very excited to be here. I am coming at you live from the state of Iowa, very different from where this thesis research was done. I guess you could say I'm in a sea of cornfields rather than an ocean now. But I'm very excited to be here, and I have a challenge for you all throughout this talk. There is a bit of irony for me being in Iowa right now, and slide 6 might help you figure that out. So today I'm going to be speaking to you about my research at the Tranquility Farm shell midden, which is in Gouldsboro, Maine. And I completed my undergraduate research and my master's thesis research at that location. Specifically, I was trying to figure out the seasonality of use at that site by using faunal remains, or excuse me, mollusk remains. Now, in case all of you are not familiar, a shell midden is, I feel like the Maine midden minders does a very good job of describing a shell midden. They're basically a cultural space located on any coastline, not just in Maine, that is created by indigenous folks over the course of thousands of years of occupation. The shell midden pictured in this slide is one of the largest shell middens north of Georgia. This is shell middens from Damariscotta, primarily made of oyster shell. The midden that I was looking at was much smaller than this. Tranquility Farm shell midden was first excavated by Warren K. Moorhead in the 1930s. His collections and findings are at the Abbey Museum in Bar Harbor, and the Abbey Museum continued on research at that location in the 1990s and the 2010s through the form of field schools. The Tranquility Farm Field School was the first archaeological experience I've ever had, so I kind of owe my career as an archaeologist to Tranquility Farm. Some research that had been done prior at Tranquility Farm before I started doing some research there. There's some carbon dates dating to about 1,240 years before present. And there were a couple questions that the Abbey Museum was looking to have answered at this site. We were trying to figure out, when was Tranquility Farm occupied during the season, because we have an idea of when it was occupied temporally in other ways. And the way we did this, and the importance of understanding site seasonality, we want to have a better understanding of people's orientation on in relationship to Maine's landscape. Shell middens are a really unique resource throughout Maine and other regions. They preserve artifacts that archaeologists get very excited about, things that wouldn't normally preserve, such as bone or wood, things that can tell us about Maine's past people and present people, but also tell us about Maine's climate history. These shell middens are threatened by coastal erosion, so being able to work on these middens is very important, because they might not be around in a couple hundred years, even 50 years. Little bit of background research. We have an understanding of when these coastal sites were occupied based on historical references. People occupied these sites, were on the coast in the summer, and then moved inland in the wintertime. I was specifically looking at the oxygen isotopes extracted from modern and archaeological soft shell clams to assess the season of occupation at Tranquility Farm. For those of you who might not be familiar with oxygen isotopes, basically the clams are marinating in the water, and as they're living their happy clam lives, they're incorporating minerals and other elements from the water into their shell composition, one of these being oxygen isotopes. The ratio of oxygen molecules within their shell depends on the sea temperature. So by measuring that ratio, we can determine what the sea or the water temperature would have been at the time of the clams harvest. So I did this research in conjunction with another researcher who is also on this call. Very excited to connect with her again. And we were trying to do something that we hadn't found represented in research literature before. We were trying to look at modern clam samples that were collected throughout a calendar year. So we collected, I believe it was like 10 samples, 10 living organisms that we then processed and made not living. And to figure out what a clam's isotopic life cycle looked like throughout a calendar year. We then chose a sample of archaeological shells, me from my study area and Emily, my research partner, from other shell middens along the coast of Maine. And we sampled those shells to see if we could match when the archaeological shells kind of fit within our modern life of a clam isotopically. We completed this research at the University of Iowa in the oxygen isotope research lab. And to do this, we basically took the very final growth margin of these organisms from the archaeological and the modern organisms and sampled that and processed it to have the isotopic readings. And we specifically sampled the outer edge of the shell as well as the chondrophore. Our results, after creating some ranges and doing a little bit of statistical analysis, primarily fell within the late fall, winter, and spring measurements. To do this and to come up with these findings, we basically created a range for the modern shells that we see here on the left. This is the modern range, and then we placed our archaeological samples within those modern ranges. And I think the thing that's particularly apt about this research is this is confirming what we have seen from historical records as well as from ethnographic research. I haven't continued this research. It kind of ended with my master's, but Emily also completed her master's research on this topic. And at the time, we were interested to see where this research went. Since our master's theses have been completed, another group of researchers, Leslie and Heinrich, who are researchers from University of New Brunswick, actually applied the research that we have done and successfully published a paper on a site in down east Maine. And so it's very exciting to see the research that we had spent our master's creating, put into use. So thank you all to my contributors and my advisors, as well as my funders. And thank you again for having me here today. I'm happy to answer questions. And thank you very much.
Great. Thank you, Kate. And we're going to have each presenter, and we'll kind of group questions as we get to the end so that each person has a chance to kind of give the context. So we're looking at the real diversity of information that can be gained from these humble critters, the soft shell clam. All right, Sarah, I'm going to pass it off to you now to talk about your research while you're at Maine Maritime.
All righty, let me get this screen sharing going. And there we go. Okay. All right. Can everyone see the presentation? I think it's up there. Okay. Well, first of all, thank you to Julia and Haley and the Wilson Museum for making these programs possible. I think this is such a great opportunity for us to have such an interesting conversation about soft shell clams. So as Julia mentioned, I did my research at Maine Maritime Academy through the School of Ocean Studies back in 2013. It was my senior research project. And my project focused on sea level rise and how it would impact Wabanaki shell midden sites in Frenchman Bay. So a little bit of overlap with Kate's research as well. And so my project, I created a vulnerability index using a geographic information system program to map the location of the shell middens within Frenchman's Bay, which is just off of MDI, to determine which shell midden sites were at the highest risk of being lost or damaged due to rising sea levels. So Kate covered this a bit already, but I'll just reiterate. Shell middens are large or small piles of shells, and in Maine, significantly the soft shell clam, that were remnants of the Wabanaki tribes over thousands of years. And up and down the Maine coast, there are about 2,000 shell middens that we know of. And even though these may seem like kind of ancient debris piles or trash piles, as the saying goes, one person's trash is another person's treasure. And as Kate touched on, shell middens can really provide us with such crucial and valuable information. So it's important that we protect them and preserve them. So a couple of the features that make shell middens important are their alkalinity. Basically the shells, other bones, or even cultural items that were buried within the shell middens. Because of the alkalinity levels, these help preserve the items that were buried within them. So we can get a glimpse at kind of the daily life of the Wabanaki people over many, many years. And so one finding from some of the Frenchman's Bay middens, I think from a 2011 study, was that over time, we found that codfish in the area were historically much larger and much more predominant. So that could be an indication of, changing climates or overfishing. So that's one way shell middens can help us learn about the past. And then also due to their stratification or their layering, the shell middens can provide, as Kate said, an environmental record or timeline of how water temperatures, salinity levels have changed over time by studying the shells and seeing those oxygen isotopes. So for my project, I was very fortunate to get assistance from the Maine Historic Preservation Commission, specifically Dr. Arthur Spees. He was a huge help in my research project. And I utilized ArcGIS, which is a mapping program, to use different mapping layers to see where these shell middens were located in Frenchman's Bay, and then to add different risk variables to the shell middens to see how vulnerable they were to being disrupted or eroded due to sea level rise. So the risk variables I utilized were damage to the site that had already been recorded, the slope of the coast where the site was located, soil type, the elevation of the site, coastal bluff hazards, hurricane flood zones, and the distance of the site to the shore. So for my hypothesis for the study, I thought that we would find 50% of the 99 studied shell middens would be facing a high vulnerability of being destroyed or damaged by changing coastlines. And with the mapping data and ArcGIS, I created an equation that would result in the shell midden vulnerability index. So the ratings were applied to each of the sites, and the priority level basically just indicated which sites should be excavated or given, upfront attention due to being very vulnerable to erosion or destruction. So fortunately, at the time of my research in 2013, my hypothesis was a bit higher than the actual results. So the results indicated that only 6 of the 99 shell middens in Frenchman's Bay were considered high risk due to their vulnerability index. So they had the most risk factors indicated on them. And out of those 6, only 1 of the sites had not yet been excavated. So that would have been considered the highest priority site. And as you can see, the highest sites are indicated by the red dots. They're all kind of in a similar location around Flanders and Sullivan Harbor. And as you can see with the yellow, there's a bit of the low to moderate, or excuse me, the moderate to high risk sites as well within that same area. So my hope with this study not only was to kind of connect, science with history at Maine Maritime Academy. It was a little bit of a unique research project for MMA, but I thought it would be a great tool for future researchers to utilize, just kind of a stepping stone within the preservation process of these shell middens in Frenchman's Bay. So it's been a while since I looked at my research, so I appreciate the offer from Julia and Haley to be a part of this. And in my preparation for this talk, I was really excited to find out about the Maine midden minders at the University of Maine. As Kate mentioned before, it seems like an amazing program where people can really get involved with the preservation process of these shell middens. They can, they have a great website with lots of resources and information on there and how people can get involved. So although my results didn't, they weren't very shocking or extreme, it shouldn't take away from the importance of these sites. Even low priority sites can still possess some risk characteristics. And of course, as we know, sea level rise is still happening. So shell middens are at higher risk every year. So with these groups like the midden minders, it's something that people can really get involved in if you're interested in it and learning how to protect this really important cultural resource. Of course, you can also visit the whaleback shell midden sites in Damariscotta. But if you take anybody, make sure you do it safely and respectfully so that we can all be a part of preserving these middens. And then I have to put a little bit in here for Dr. Spiess's book, The Turner Farm Fauna. If you're really interested in this subject, I would highly recommend reading this book. It's very thorough about their findings at Turner Farm, which is on North Haven. And it's just a beautiful, they've got beautiful descriptions and imagery of their findings from the Wabanaki people in that area. So thank you again to MMA, my professors, Arthur Spiess, the Maine Historic Preservation Commission for getting me through this research project and helping me with all the information gathering for it. Thank you.
Wonderful, thank you, Sarah. Yeah, and it was really fascinating to see Sarah's work in the context of Maine Maritime Academy. So definitely a different approach there. Sarah, if you want to stop your screen share. All right. And so now we're, again, as I mentioned in my introduction, we're going to jump ahead a fair amount to Sophie's work, but in the same landscape, the same geography. We're looking at how incredibly important this organism of the softshell clam and the harvesting of clams continues to be to the people who make a life and living around Frenchman Bay. So I'll hand it over to you, Sophie.
Thank you so much. And thank you for the opportunity to be here today and to chat about clams. Let me just pull up my presentation super quickly. And alrighty. So hi, everyone. Thank you so much for being here. I'm Sophie Shivers, a sophomore at the College of the Atlantic, majoring in human ecology with a focus on marine science and environmental policy. And I'm here today to talk a little bit about softshell clams and the softshell clam industry, mostly through the lens of the work that I did this summer with the Schoodic Institute and the town of Gouldsboro, and also some of the work that I've done as a research assistant for the college. So none of this is really my data or my research, but it's research that I've been able to participate in and assist with. And so I'm super excited to get into it with you guys. So we've been spending the last half hour, and we'll continue to spend a little bit of time with these guys. So our little Maine softshell clams, talking about the Maine fishery that relies on them, the threats facing them, and what towns are doing to mitigate those impacts. So these are softshell clams. Their name comes from their calcium carbonate shell that's a little bit chalkier than your average mollusk shell. So they're more brittle than like a quahog or a cherry stone. So how many of you guys have ever been clamming? Has anyone ever been out to the flats? Awesome. Looks like a couple folks. That's super sweet. So there are two super common techniques for removing clams from the mud. And I'm going to show two super quick videos from Dr. Bridie McGreevy's resource ClamCam. She's a professor at the University of Maine and has worked with fishing communities to sort of create a little more pride in the community itself. So this is a video of, clam digging, where the clammer sort of digs into the mud with a large metal rake or hoe and then pulls the clams out from the mud by hand after sort of uncovering them in that way. The other technique is called pulling. And it's a little bit less common, and it can only really be done in super soft sediment where the clammer can sort of stick their hand into the mud and pull the clam out by hand. Both techniques require a lot of strength and practice. I've tried both. Digging requires a lot of strength. It's really hard work, and I broke more clams than I harvested, to be entirely honest. And we can just say I didn't see a single clam while I was out pulling for them. So now let's talk a little bit more about the fishery on the whole. So according to the Maine Department of Marine Resources, the primary authority on Maine fisheries, softshell clams are the second biggest fishery in the state according to size. They're that little, or according to value, they're that little red triangle there. So it's also, because fisheries are such a major source of income for the state of Maine, the clam fishery is important in a broader context than just clamming communities. It's also a unique and important form of income for many communities. The cost of entry is low compared to many other boating fisheries, and the time commitment is a little bit more flexible. So people just need sort of a license or commercial license, boots, a clam hoe, a bucket, and a ride to the flats in order to sort of participate in the fishery. So it's a really accessible way for people to have independence and self-determination over their employment. So the issue right now is that the fishery is rapidly declining, as you can see from this graph here. The most recent fall sort of started in the early 1980s. And one of the most likely reasons for this is climate change. So we usually talk about climate change in one of two ways when we're talking about clams and shellfish. The first is ocean acidification, and the second is warming. So acidification is certainly a concern for shellfish. Their shells become harder to make under acidic conditions, and they can often warp. But it seems like this really isn't the primary driving factor in the declines over time for clams. It seems much more likely to us that warming is the concern. And Maine is the Gulf of Maine is one of the fastest warming bodies of salt water in the world. And so this is a graph from Pershing et al.'s 2015 study. My apologies, it's a little bit hard to read, but there's a pretty strong spike in temperature in the 1980s, which correlates pretty strongly with the beginning of that decline for the clams. So this sort of begs the question of why is temperature such a concern for this species? They live as far south as North Carolina, so they are resistant to warmer temperatures. The primary threat that we talk about is the European green crab. The crabs are much more successful. They're a pretty dominant predator of the clams, and they're much more successful when winter conditions in particular are warmer. They're able to have larger populations and reproduce more, so have more kids when these temperatures are high. So how do we know that these guys are sort of the primary threat to clams in down East Maine? So most of the research that I've done at College of the Atlantic has been with Dr. Chris Peterson, contributing to a growing body of predator exclusion data. We're part of a coast-wide shellfish monitoring network that uses similar materials and protocols to gather information on clam larva arrival and mortality due to predation. So I want to talk for a second about the methodology we use to determine the impacts of predation on this fishery. So up here in the left corner of the image is a Beal box, which was named after Dr. Brian Beal, whose name is synonymous with mudflat research in Maine. He designed these boxes. They're 1 by 2 foot boxes covered with pet screen, and the screen is just big enough to allow clam larvae to settle into the box, but doesn't allow crabs or other predators to enter. So we use these boxes to be able to create sort of a predator-free environment for the really young clams to grow up in. We typically put them out in the spring and pick them up in the fall. And when we pick them up, we also take cores of the adjacent mud to get a comparable sample of mud without that sort of predator exclusion treatment, as we call it. So, and then us lucky undergrads get to spend about 2 to 4 days counting thousands of tiny clams. That image up there in the top right is a tray full of all of the clams that came out of that Beal box. That was a good year for recruitment. So what we're seeing is that we're finding orders of magnitude more clams in boxes than in the mud. So that's telling us that clams are getting to the flats, and you can sort of see in this data here, the cores are the orange, so that's the mud that doesn't have any kind of predator exclusion treatment, and the green are areas with predator exclusion. And so that's telling us that the clams are getting to the flats. They're arriving and they're surviving. They're just getting eaten really, really quickly. So this looks pretty bad, but we still have a pretty major fishery. So what are towns and people doing to keep the fishery alive? My work in shellfish and in Gouldsboro has primarily been around this sort of work. So I spent this past summer working with the Schoodic Institute and the town of Gouldsboro, a down east town on the Schoodic Peninsula, focusing on a new kind of resilience effort. So shellfish is a really interesting area of fisheries management because the state and municipal governments share management responsibilities. So this means each community is allowed to decide to a large extent how they want to manage their resource. So a common tactic to boost clam populations is to seed the flats. That sort of means taking year-old clams and putting them out on the flats under nets for about a year. This reduces mortality within the first year and protects them from crabs while they grow. Anywhere from 1 to 4 years later, depending on the cove, clammers can go out and harvest the clams and sell them to market as they would any sort of other wild caught clam. So this is great. It's sort of keeping the fishery afloat, but it can be super costly for towns as the year-old clams are coming from a hatchery and that someone's put a year of food into those little clams, and so it can be kind of expensive. So in Gouldsboro, we were trying to see if there was a way for us to reduce that cost. So thus the Shellfish Resilience Lab, affectionately called the Clam Shack by some of us, was born. So the idea of this was to take clam spot, which is sort of very newly metamorphosed clams, which is about the size of a grain of brown rice or smaller. And so you can collect that in the intertidal, or you can buy it fairly cheaply from a hatchery. So that spat is then taken and put in nursery trays, which are kind of giant floating Beal boxes, and you can see them in the image right above that number 2, or they're put in tanks inside of the lab to feed and grow over the summer. In the winter, all of those young clams are collected from the trays and tanks and placed in cages and bags to do what we call overwintering. So this just means cold water circulates through and they hibernate. And then in the fourth year, or in the fourth step, sort of after the winter period is done in the spring, they're a year old. They're seed clams now, so very similar to what would have been purchased from a hatchery. And they're like maybe the width of your pinky finger, and they're ready to be put out onto the flats under nets. So the project is still in process, and it's just one example of how municipal shellfish programs are getting creative and developing new methods to preserve their wild harvest fishery. So to sum up, climate change is undoubtedly affecting softshell clam populations in Maine. There's a lot of data demonstrating the impact of green crabs on the fishery and the impact of temperature on the fishery. My arenaria is an example of a commercially relevant species that's facing the impacts of climate change. And I think these species are really important because they act as indicators or wake-up calls. We notice when something's going wrong. So thank you so much for having me today. And thank you to my supervisors over the last few years, Dr. Chris Peterson, Sarah Hooper, people from the town of Goldsboro that have made my involvement in all of this possible.
So cool. I mean, I knew that there were cool things to be learned from clams, from having interacted with Kate's work and with Sarah's work, but it's really neat to even expand that even further. So thank you very much, Sophie. So I've got a couple of questions that I'm going to put out to our panelists, and then we're going to turn it over to the audience to ask your questions. And I will acknowledge we share these with our team, with our panelists here in advance. I'm going to tweak them a little bit based on what I heard from your presentations, but not too much. So I think that a lot of folks probably wouldn't think about clams and softshell clams as as an important source of data when we're talking about understanding how Maine's climate has changed either more recently or over the past several thousand years. Starting with you, Sophie, how did you come to settle on the softshell clam as opposed to some other organism in trying to better understand what's going on in our environment?
Yeah. I came to College of the Atlantic hoping to study marine organisms and particularly sort of their relationship to climate change and the way that those things are managed. And I was super lucky that my major professor, Dr. Chris Peterson, has focused in clams and has worked with students on this fishery in the past. And so it was an easy avenue for me to sort of start doing a little bit of hands-on research and also getting to experience a management structure behind a resource like this. And I've just sort of found that they're an incredible organism, but also just sort of have an incredible group of other opportunities around them that's worth sort of staying around and observing. So that's sort of how I wound up here.
Nice. All right. So how about you, Sarah? How did you, from Maine Maritime Academy, come to do work that really focused around what we can learn from, in a certainly different way, from softshell clams?
Yeah, when I was trying to figure out what I wanted to focus on for my research project, I was really trying to find a way to combine like a cultural resource like shell middens with climate change. And in talking with my professors and a little bit with Dr. Spies, it fell in my lap a little bit because I got a lot of assistance from them, and they really taught me a lot about shell middens because I didn't have much experience studying them at MMA. And I wanted to show how we could connect a cultural resource with scientific information, and how we could learn from basically what someone considered thousands of years ago, just some debris, how we could take that today and learn how climate has changed over the years, and what we need to do to protect this resource so that we can keep utilizing it in the future. Because really, not only can we learn about water changes and things like that, but also marine life changes with fish bones that are found in it, or even other animal bones. So yeah, I was very fortunate with the advisors that I had. And with the information I was able to utilize from the Maine government to do this research. And it was fascinating to learn about it.
Nice. And I know a little bit in terms of background on Kate's research. I had the privilege of sitting on her thesis committee, so that was very cool. But I also, having worked with Kate doing some of the faunal analysis on Tranquility Farm, there certainly were a lot of different critters represented in the faunal remains. The faunal, for those of you who aren't familiar, is the remains of food, the remains of animals that people were eating at these village sites. How is it that you came to focus on the clams, the clamshells, Kate, as opposed to some of the other faunal material at Tranquility Farm?
So for my undergraduate research, I tried to look at the faunal remains and see if there was any kind of remains that we found that would be indicative of a certain season. And the sample that I pulled wasn't really indicative of any particular season. And as I had been starting to research shell middens, everybody was always saying, you gotta look at the clams, you gotta look at the clams. And I feel like after doing some additional research, very often people will take the clam and they'll cut it in half and then do like a sample along the growth bands. But nobody had really done the modern comparison with clams yet. So I think kind of finding a way to do a seasonality analysis with a new take on it and see if there's a way that we can incorporate modern samples into analyzing archaeological samples was really interesting for me, just as a way to try to answer this question of when was this site occupied, and add to the broader research of seasonal occupation along the coast of Maine.
Great, thank you. And certainly that's something that I'm really seeing out of across all of your three research projects is this the really powerful significance of the relationship between the softshell clam and the humans that subsisted off of them, and how that relationship is so telling, and how it informs across time in ways that are really linked and fascinating. That the folks of, in all three cases, the folks of Frenchman Bay and Goldsboro had a really important relationship with these little shellfish. In keeping with kind of the theme of the Connecting to Collections program series, in terms of how do these connect to what we're thinking about today in the here and now, and certainly Sophie's is really ties us into that. But what might be something that you feel that came out of your research with that could inform decisions or actions that we as individuals or as organizations can be taking today, and what can we learn about our actions today from the softshell clams over several thousand years and the people who've been interacting with them. We'll start on your end, you with Kate.
So there is a lot of research from other individuals looking at clams as indicators of, and not just softshell clams, but other mollusks as well, particularly more long-lived mollusks. I think quahogs are prime example, and that live in deeper environments as records for what temperature change was temperature change throughout the Gulf of Maine's history. So, I mean, having a really solid understanding of what the Gulf of Maine's temperature would have been like a couple hundred years ago, based on the oxygen isotopes in mollusks, is really important. There aren't too many other ways we can find records like that. And soft-shelled clams in particular, I mean, we have so many of these archaeological sites along the coast that finding a way to utilize them as a resource for understanding Maine's past temperature history and past history of clamming and utilization of ocean resources is really important. And it was exciting to be part of it. And I think it's interesting to, it's been interesting to be part of that research process.
Great, thank you. Sarah, what outcomes or things that you experienced or learned during your research help inform us about decisions and actions that we make today?
Yeah, I think, you know, a little bit echoing Kate, our research kind of cross connects in different ways, but I think just with like the groups like the Midden Minders at Maine, just bringing more awareness about these resources and how they are so plentiful up and down the coast. And kind of, I guess, thinking out of the box a little bit too, because when we think about climate change and sea level rise today, what we hear in the news and what we hear in our own studies is that, you know, yes, it will affect our infrastructure and the way we as, you know, society lives day in and day out as these temperature changes and sea level changes keep occurring. But I think also keeping in mind about these cultural resources that will be affected too. Not only shell middens, but other historical sites, you know, along different coastlines will be affected by climate change. And even if you just share this information with friends and make them more aware about it, just you know, bringing a light to this other side of climate change, where it's not just affecting our future, but affecting our past and our cultural records.
Absolutely, very cool. All right, and Sophie, again, coming back to the present, what can your research help inform in terms of the decisions that and actions that we as individuals and organizations are taking today?
Yeah, I think that one of the things that has been most fascinating for me in the process of getting to do this research is looking at the management structures behind softshell clams in Maine. And so that co-management, that shared responsibility between state and municipal level governments, I think is such an interesting and powerful way to pull community-based conservation to the forefront of statewide or national governance. And so that's been something that I spend a lot of time thinking about how that could be applied in ways to larger organizations and larger management structures. So that's definitely one of the things that I think can be really powerful for sort of helping preserve resources and make sure that they're being preserved for the people that need and use them the most.
Great, thank you. All right, I think I'm gonna, Haley is monitoring our chat and so on, but I think we want to now, I want to give folks in our audience a chance to ask some questions of our panelists. And I know we've got one chat question already. Do you want to read that out, Haley, and then we can?
Sure. So our question in the chat from our audience is, what other foods were being consumed at the prehistoric sites? Is that something that can be seen from the shell middens? Or do we know?
I think I'm going to pass that one probably to Kate. It's going to be our best. I mean, other than me answering, I'm going to pass that to Kate to answer a little bit.
Yeah, so there's we find evidence of a lot of fish and birds and mammals throughout the site. Trying to think of some fish. I'm trying to remember, Julia, was there like one type of fish that we found most in particular?
So it depends on the site. Certainly at Tranquility
Yeah.
Farm, it was a mix of them. There's certainly a number of sites where we find a lot of tomcod, frost fish or tomcod. Cod in general have a vertebra structure that is very distinct and preserves well. So we see evidence of those. We see evidence of sculpin and of sturgeon. The sturgeon evidence is really cool. Sturgeon have this exoskeleton that's very distinctive and preserves well. A whole lot of fish don't have a bony skeleton. Their skeleton is made of like cartilage sort of, and so it doesn't actually preserve, but we certainly see, and Kate mentioned the fascinating changes in the size of codfish that we see from Wabanaki pre-contact sites to present day codfish, huge drop in size. But yeah, I mean, everything that lives, all of the wild critters that live, and that fly and walk and swim were harvested and consumed by Wabanaki folks who are living along the coast of Maine. and it changed, from season to season, but there was a very, very diverse and broad use of those resources, and also of plants. Plant data isn't as well preserved and as well understood as some of the animal data, but Sarah shared the Tranquility Farm fauna book. If you really want the full catalog of what critters were being eaten by Wabanaki folks on the coast of Maine, the Tranquility Farm, the Turner Farm fauna book is the encyclopedia of that information.
Adding on to that, some other interesting species that are found in middens. At the Turner Farm site, they were finding swordfish, which we don't have easily accessible along the coast of Maine now. But I mean, they were taking down these swordfish in canoes. And so we're able to find evidence of that in the archaeological record. And then I know at Tranquility Farm and other sites along the coast, we are finding evidence of the extinct sea mink, as well as the extinct great auk. So thinking about these middens, they're some of the only records that we have of these creatures. And I think that's kind of one of the things that sparked my interest most about these sites is, I mean, we're able to see things from creatures that aren't around nowadays.
Wow, that's really interesting. I'm glad that question was asked. So thank you, Mark. And we have another question in our chat. It is, what caused the decline in clam population from the late 50s to the end of the 60s, and then spiked back up before declining from the late 80s to now? So what caused the decline in the clam population?
Yeah, so that first dip in the graph is also associated with a series of really warm years. And I think also a large spike in green crab population. But my temperature graph didn't quite go back that far.
Interesting. Thank you.
Do we know when the green crab arrived in Maine coastal waters?
I think they've been here for several hundred years. Just sort of the increase in temperature has really caused their populations to blossom.
Yeah,
And then Dr. Peterson just clarified that for us, the early 1800s.
So not something that Wabanaki folks were contending with for their clam harvests. All right, folks are welcome to put additional questions in the chat. You're also welcome to either raise your hand or unmute yourself and ask questions.
So I have a question for the archaeologist in the group. So this is Chris Peterson. So it's a taphonomy question, really, a preservation question. So it's always struck me that like 6,000 years ago, that the second most common fish in shell middens were swordfish, which just kind of blows my mind that people were landing these in canoes. To what extent are big bone things like cod and swordfish going to differentially preserve in shell middens, and to what extent do they end up getting overrepresented because they just preserve better versus, you were saying, Julia, that cod have really distinct vertebrae, and so my question's kind of a differential preservation question about what some of our data say and how it could have some potential biases in it or not.
I think there's definitely going to be a bias. Even just thinking about the screens that we use for analyzing these samples. I mean, if we're looking at for smaller fish, those fish vertebrae are often smaller than our 1/4-inch screens that we're using. So even if they did preserve, they might not be something that we actually find as we're doing our excavations. I haven't spent as much time with the fish, so I can't speak entirely to how well they preserve. But I do know that we will find oftentimes little pockets of very small fish vertebra. Sometimes we're not able to identify the species. I'm sure somebody who is an expert in fish would be able to. But for those, often I remember we would switch to dental tools and trying to preserve at least a sample to make sure that that individual was recorded. And But I think oftentimes it's like, this is a problem with any sort of archaeology, is our screen sizes often dictate what we're finding. So smaller things might not necessarily be preserved. But that's one of the important reasons why we take soil samples. So taking a bulk soil sample from the site, which I do remember doing at the shell midden digs I have worked on, is a great way, so we can do flotation or other sorts of analyses to try to capture what we might be missing in the 1/4-inch screens.
Yeah, certainly another key area of species that aren't represented in these, even though the shell midden preserves a lot because of the alkalinity of it, we're not seeing like lobster and crab shell that doesn't preserve. So that's obviously key resource. I was target one of our audience here, Arthur Anderson, who's still working, who's actively doing research along the coast of the Gulf of Maine. Do you have any thoughts on that in terms of, are there ways that we can better understand the species that aren't being represented, that we're not seeing in the archaeological record?
Yeah, I mean, I think as Kate said, the screen size is a big one, and thus taking samples that we can really go through to get a sense of what might be there, and sometimes even some numbers that we can try to apply more broadly, even though the shell middens are so variable. But yeah, that detailed sampling, column sampling really helps. But I think also in thinking about the types of marine environments, because these shell middens are often right in front of a clam flat. But when we get a more detailed look at things, we see that people are sampling some different marine environments and bringing them all back to the same place. So if we can get a sense from the animals in the midden, what other environments people might have been exploiting, we can think about what might be there that wouldn't be preserved in the situation that we have. So those are ones that come to mind, but yeah, the screen size is a good one. And absolutely that bias in the preservation towards big things could also be a bias towards hunting big things, but the preservation bias is a big one.
Thank you for being a willing audience participant, as I recognize folks in the audience. So Haley has another question from our audience that she's gonna share with our panelists.
Yes. And before I read that, I just want to note that it's really fun to watch the people who I can see on screen, and to, as Kate talks, to see her support from her research partner, Emily, like as she answers questions, she nods along. So it's really nice to have such an inclusive audience. So this is really fun. So our question is for Sarah. Sarah, did you find in your research that any of the factors were more significant in predicting risk than others?
That's a really good question. I think, going back between the mapping layers and things like that, probably the highest kind of risk factor was the distance to the shore. If it was closer to the shoreline, then it would have been for sure at a higher risk of being affected by rising sea levels or erosion due to higher wave action. So probably up there would have been the soil type as well. If it was on a more rocky bluff and kind of protected from the wave action, or if it was in like a looser, sand or gravel area, then it definitely would have been at higher risk of being affected by wave action.
Great, thank you. Other questions? Sure, Don, go ahead.
Hi, I'm Don Tenney. I live in Castine. And I have a question about the green crab. It's probably a question for Sophie, the green crab and the clam age. I'm a sea kayaker, and down on Ecomogan Reach is a lovely little island there that looks almost like it belongs in the tropics because it's got a gorgeous white beach, but the beach is not sand. It is all crushed clamshells. And down at the low tide mud layer, there are more whole clamshells, all dead clams. And I'm assuming they were all victims of the green crab. So I'm just trying to confirm whether it's a case that the green crab can eat clams of any age and they can excavate the mud and get clams further down, or that they just basically clean off the spat from the surface and prevent them from penetrating into the mud layer and establishing themselves.
Yeah, absolutely. So I think what we usually see is that small crabs eat small clams and large crabs eat large clams. So I think they typically sort of will find a siphon and grab it with one claw while digging it out with the other. And they're very effective. I have a shell here that sort of, it has a little break out of it, and green crab predation can often look like that they actually are able to break the shell and remove the clam. And so I think it's hard to tell sometimes if a shell has just been sort of battered on its way to a Maine coastline and the clam died for other reasons, but often they will leave pretty characteristic marks of predation, and they are really effective predators. But the little crabs definitely do clean off spat pretty effectively, but big crabs are also able to eat pretty sizable clams. Yeah.
Yeah, it's quite a striking thing to see when you see
It is.
that many clamshells all in one place. Yeah, thank you.
For sure. Yeah.
Sophie, can you hold that a little closer to your camera? Can we have another look at it?
Yeah. Is that in any kind of focus?
Yeah, that's closer. Yeah. All right. Thank you.
Yeah.
Any other questions popping up for folks? We've got these terrific women on the line here.
This one's for Kate. Have you seen any signs of, of predation in archaeological clams?
There were some where I'm not entirely sure what organism did it, but they were, maybe this is something that Sophie can answer. There were drill marks. Is that from a worm of some sort? It was kind of usually towards the not on the edge of the clam, but more towards the middle chondrophore area. Is that a worm or something?
The two other, and I'm really not super familiar with sort of prehistoric or older sort of predators that were here, but moon snails drill holes, pretty nasty holes using their radulas. And I think certain worms may also be able to drill holes. But yeah, I think the moon snail would probably be the most likely culprit.
I see Pam also dropped that in the chat, so it seems like we have 2 votes for moon snail right now. But I definitely noticed those in our samples and was thankful that it wasn't near the margin that we needed to be sampling.
Yeah, I've seen those on Saco Bay too, and it's always been a little bit of curiosity in the back of my brain, and I'm wondering how diagnostic the signs of predation on the archaeological clams might be, and whether that would be at all interesting. Speaking of proxies and speaking about other animals that don't preserve and all of that jazz, but maybe
absolutely.
Maybe I'll think more and send it.
Yeah, we'll follow up.
Any other questions? All right, well, I'll wrap us up by once again, a huge thank you to Kate and Sophie and Sarah for sharing your research. I know for Kate and Sarah, I was asking them to dig back a little bit into things they hadn't worked on super recently, but really fascinating work. And just again, to me, fascinating to see how the presence of and the relationship of an organism and humans and how they've interacted and how our actions have affected each other is just really fascinating and really neat to just learn so much through something that seems relatively humble. And you all will maybe think differently about the next pint of clams that you get when you go to Crosby's, but nonetheless, really, really fascinating. And certainly wrap up with a huge thank you to Bangor Savings Bank again for making this program series possible. And Haley, do you want to give a little pitch for the next program in the Connecting to Collections program series?
Yes, I do. Our next program in the series is on Monday, May 9th with Dan Sandweiss, and we will be looking at what we can learn from shells as climate clues, specifically Peruvian shells. And that's a nice tie because the museum has some Peruvian artifacts and shell artifacts. So that's a nice dual connection for us. So that'll be Monday, May 9th, in person, but also on Zoom. So we'll be sending out information for that soon. And if you're interested in getting a direct link, you can sign up for our e-newsletter because we send out information on our programs on our e-newsletter. And you can also email info@wilsonmuseum.org or find our information on our website.
Wonderful. Thank you all. Thank you for really fascinating conversation. And we look forward to seeing as many of you as possible at our future program series. Have a good evening.
Thank you.